discussed about glycolysis being the transformation of glucose into two pyruvate molecules. Now, if in the cell there is oxygen, then the cell can undergo a reaction known as aerobic respiration, in which the two pyruvate molecules formed are transformed in are transported in the mitochondria, where they are transformed into two molecules of acetyl-CoA, a molecule known as acetyl-CoA, by a process known as pyruvate decarboxylation, using an enzyme known as pyruvate decarboxylase. Now, the two acetyl-CoA molecules that will be formed will now enter the citric acid cycle, which is also known as the Krebs cycle, which is also known as a tricarboxylic cycle or TCA cycle, as you can see in this diagram. So, citric acid cycle is a series of 8 steps. The entire purpose of the citric acid cycle is to basically take the energy that is stored in acetyl-CoA, the molecule that is formed from pyruvate, transfer that energy in form of high energy electrons to the electron carrier molecules known as NADH and FADH. Now, the citric acid cycle does not directly form ATP molecules. It does, however, form NADH and FADH too. Now, these two, they will then be used by the electron transport chain to synthesize ATP molecules. So, on the diagram shown here, this diagram is only showing one reaction for one of those acetyl-CoA molecules formed from pyruvate. So that means the cycle takes place twice since we form two pyruvate molecules, two acetyl-CoA molecules, two citric acid cycles for one glucose molecule. So let's begin with step 1. In step 1, we take our acetyl-CoA, mix it with a 4-carbon molecule known as oxaloacetate to produce a 6-carbon citrate molecule. Now we take the acetyl-CoA and get the acetyl portion, leaving out the CoA portion. The CoA portion is coming to be used at a later stage of this reaction. So we are only getting the acetyl portion, which has 2 carbon atoms, and we combine it with a 4-carbon atom known as oxaloacetate using an enzyme known as citrate synthase. In the end, we have a molecule or a product known as citrate. Now, when you look at this citrate is going to be used in a reaction called a decarboxylation reaction. But before that happens, that citrate has to be restructured. Why? When you look at this hydroxyl group, it's different from the hydroxyl group in the position in which isocitrate, the product formed from citrate. Hydroxyl group is different from that of citrate. So what we have actually done here is that we have restructured the citrate, converting it into an isomer known as isocitrate. The only difference is that the hydroxyl group in citrate is in carbon 3, the hydroxyl group in isocitrate is in carbon 2. This is done so as to prepare the citrate molecule for a reaction known as decarboxylation reaction. The decarboxylation reaction is a reaction in which a carbon dioxide is kicked out of the molecule. Not only do we have a decarboxylation reaction, we also have a dehydrogenase reaction. This means a hydrogen will be removed from isocitrate and it will be given to NADH, or rather NADH, NAD plus is abstracting a hydrogen atom, it being reduced in the process. At the same time, the carbon dioxide is being kicked out. Now that we have kicked out the carbon dioxide from isocitrate, we remain with a 5 carbon atom because one carbon atom has been kicked out in the form of carbon dioxide, and this process was taking place under the guidance of isocitrate dehydrogenase, an enzyme which catalyzes the abstraction of the hydrogen from acetate. We now remain with 5 carbon molecule known as alpha ketoglutarate. Now, in step 4, the alpha ketoglutarate further undergoes a decarboxylation to release a carbon dioxide, and that means we go from a 5 carbon to a 4 carbon known as succinyl-CoA. So moving from a 5 carbon molecule to a 4 carbon molecule known as succinyl-CoA, and this is catalyzed by alpha ketoglutarate dehydrogenase complex. Now, take note that this is a decarboxylation reaction again. At the same time, it's a dehydrogenase reaction. So carbon dioxide is being kicked out, NAD class is being reduced, and this NADH, remember the purpose of forming this NADH is so that this NADH can transfer these electrons to the electron transport chain. So now that we have succinyl-CoA, we are going to take part in a reaction known as formation of ATP, or rather GTP, which is an equivalent of ATP because it's going to be converted into ATP. So now the formation of GTP involves a nucleophilic reaction. What I mean is that there is a CoA part on this succinyl-CoA. Now where is this CoA part coming from? This CoA part came from pyruvate. Remember we said the acetal portion of pyruvate was removed, and that acetal portion was the one which reacted with the oxaloacetate. Now, the remaining portion was the CoA part. That CoA part in step 1 is the one which is going to be attached to alpha ketoglutarate to form succinyl-CoA. So that's the CoA that we're seeing here. So now succinyl-CoA, the CoA part is replaced or substituted by a nucleophile known as phosphophosphate. It's clearly present in the matrix of the cell. So it's going to kick out the succinyl-CoA, the CoA part, and this molecule will become a succinyl phosphate. Now this phosphate is the one which is going to be transferred to GTP to form a GTP. GTP simply means guanosine diphosphate, means it has two phosphates. So when you add this one phosphate from succinyl phosphate, it's going to become guanosine diphosphate. And the molecule that we have now remained with is succinyl. Now we have formed our succinate here, as you can see, and our GTP has been formed. This GTP is going to be converted into ATP. So this is an equivalent of an ATP molecule. So just same as forming an ATP molecule. Now succinate is going to be mixed with FAD, and FAD is going to extract 2 electrons from succinate under the guidance of the enzyme succinase dehydrogenase to form fumarate with a double bond in between. And in the process, we have accepted 2 hydrogen atoms or 2 hydrogen ions or indeed 2 electrons. These electrons are going to be used in electron transmission as well. So now FAD has already taken part in the reaction of converting succinate to fumarate. Now after we have formed fumarate, we input a water molecule to form marate. The purpose of converting fumarate to marate is so that we can go all the way and regenerate the molecule that we started with, which is fructose acetate. So at this step of things, continue running its course. So we form marate from fumarate by inputting a water molecule, which puts a hydroxyl group there and breaks the double bond. Now after forming marate, we are going to use an electron abstracting molecule NAD plus, which is going to abstract the hydrogen and to release another hydrogen so that we can go back to forming regular acetate and the cycle begins again. Now, remember, the purpose of this reaction is to form high energy reacting carbon, which is NADH and acidic. Now, when you look back, we carry these two molecules of acetyl-CoA. But this diagram is only showing for one molecule of acetyl-CoA. So this means whatever we have produced here, whatever number of energy plus and Kb plus or Vtp plus we produce, we're going to multiply that by 2. So we have produced 1, 2, 3 molecules of energy H. So we're going to multiply that by 2. It's going to give us 6 energy H molecules. And we're also going to get 2 FAG molecules and 2 Vtp molecules, which are equivalent of 2 ATP molecules. So that's just like the citric acid shape or the cycle, the citric cycle, or the Phillips cycle in a nutshell.